three panelists. So John helped setting up the meeting and the registrations. So he will also record the lecture and share the recordings with all of you after the lecture. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. So Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. Also manages the Purdue side of the Schematic core cancer bioinformatics, which is a joint core for cancer researchers at Purdue and Indiana University. She has a background in biochemistry, molecular biology, and computational biology. So her research interests include single-cell analysis and data integration. So now I will hand it over to Nadia.
Hi, so thank you, Min. Welcome back. So we're glad you're here. And again, another few housekeeping notes. As per usual, we will keep you muted throughout the duration of the seminar. And if you have any questions, go ahead and use the Q&A function that you see at the bottom of your Zoom screen. And then we will take questions after the seminar and possibly in the middle of the seminar if we have enough. So if you have any issues, just go ahead and message me. And again, for questions, use the Q&A function. So thank you, and I'll hand it back over to Min.
Thanks, Nadia. So it's my great pleasure today to introduce Dr. Constantine Gatsonis from Brown University School of Public Health. So Dr. Gatsonis is a Henry Goddard University professor and also the founding chair of the Department of Biostatistics and the Center for Statistical Science at Brown. So his research covers a wide range of evidence-based diagnostic medicine, addressing both the methodology development and subject matter with a focus on radiomics and the comparative effectiveness of the screening and diagnostic modalities. So we have been talking a lot about like genomics, transcriptomics, proteomics, metabolomics yesterday in cancer research. So it'd be really nice to know how the radiomics can be used in cancer screening, diagnosis, and prediction. So Dr. Gatsonis is a statistical consultant for the New England Journal of Medicine. So he was educated at Princeton and Cornell, was elected fellow of the American Statistical Association, and received a long-term excellence award from the Healthy Policy Statistics section of the American Statistical Association. So with that, I would like to welcome Dr. Gatsonis, and he will be speaking on the machine learning methods in diagnostic imaging.
Hello, and good afternoon. It's afternoon where I am in Boston, and we're still basically indoors here. I hope you are more outdoors than we are. When we last had this lecture, that was 2 years ago, it was in a nice big auditorium at Purdue. It was warm outside. It was very pleasant and collegial. So this is a slightly different setting, but I guess we all have to learn how to move along. What I will be talking about today is another kind of omics, and that's radiomics. And this is now the application of machine learning in diagnostic imaging. So we have a very interesting situation there, namely the sky is the limit in terms of markers that one can generate by using radiomics in the analysis of images. There is a lot of difficulty in kind of moving these markers to clinical settings. So the translation from what radiomics can produce to the clinical setting is a fairly challenging exercise. And so what I wanted to discuss today is first, give you an overview of the kinds of markers that are being produced in that space. And the focus is going to be on markers which are useful for eventually for clinical care or for clinical trials. I will emphasize deep learning because this has by now getting a fair amount of attention in the analysis of imaging. But I will also discuss feature space analysis, which is a more traditional analysis in radiomics. I don't need to convince you, obviously, that this is at the center of a lot of attention by researchers. As much as there's growth in biomarker research, and these are some plots of how many papers came out in biomarkers or markers in the last, say, 40, 50 years. They're going up to maybe 50,000 a year and so on. But there's a similar growth in radiomics. So you can see here in this graph, it used to be very little, but by 2019, end of last year, there were maybe 800 or 900 articles with radiomics in title and abstract, and this number is really skyrocketing. There's a lot of interest in radiomics, and for instance, the Journal of Radiology, which is the official journal of the Radiology Society of North America, they started a special issue. I'm not sharing my slides, it says here. Is it coming out?
Yeah, I can see them.
Okay, I'm sorry.
You have it on the presenter mode, so you need to switch to your other screen.
So do you have me on the other screen now? Can you see the slides with the graphs?
Yeah, but we need another screen. Like you probably have two screens. So this is the.
Okay, just a sec. Let me see.
That looks good.
This is great.
Okay. All right. Thank you. I haven't said anything that needed slides so far. So these are the graphs that I wanted to show you about the growth in radiomics. And before that, there was the graph showing 50,000 papers a year in biomarkers. So what is radiomic analysis? So we have two kinds of radiomic analysis, if you wish. If you look at it from the methodologic perspective, one is analysis in feature space, and this is explained mostly here. I'll explain that first, although my emphasis is going to be on deep learning. The reason for this is feature space is more like other high-dimensional data analysis that goes on in biology, in genomics, proteomics, etc. So in feature space, the way this works is that you have an image, and that image is used to generate particular features, which are essentially summaries of aspects of the image. So they could be features based on signal intensity, or more complicated features like edges, or description of textures. This is one of the most complicated features. So all of these features are summarized as, are generated by computer programs that go through the image and do the computations. Then they are summarized as high-dimensional vectors for a particular, for an image. And then people use approaches that you have seen probably from other kinds of omics, classification, nearest neighbor potentials, support vector machines, random forests, and so on, to make various predictions and classifications. In order to come up with these features, you need to have some pre-processing, and that means you need to identify the region of interest. You need to identify, a contour around it and so on, and you may even go into three-dimensional versions of this. But generally, feature space analysis then is, as described in this side of the graph, is kind of very similar to what is done in other areas with very high-dimensional data. So that's the feature space analysis. The other kind of analysis is via deep learning. And deep learning is kind of diagrammatically depicted here. And I will go into a little bit more detail here. So deep learning starts with a set of inputs, and these are the input layer, as it were, in other words. So you could think of an image, and every pixel or something would generate an input here. And then it has neurons in the inside, which are called hidden layers, which take the image, which take the input, and they go through an algorithm and produce a certain output. The output then is taken by further neurons in the next layer and so on, and they are connected with each other, and every neuron communicates with all others, etc., depending on the architecture of the neural network. And then at the end of the day, the hidden layers are over, and then we go to the output layer where all of the information is combined, and then we get the output from the neural network. Now, this is obviously an extremely high level of description of this, but roughly that's the process, as it were. And when it comes to images, the deep learning networks that we use have some particular flavor to them. You can see it here. You start out with the input layer, and then there are some layers which do the so-called convolution. Convolution is a way of local averaging of the images. Because images have a spatial structure when you see it on a two-dimensional version. And so that's what the convolution layers are trying to make use of. And then they go to a maximization layer, and then they can have more convolution layers and so on and so forth until you go to the end where you have your output layer, and that you can feed into various kinds of classification algorithms. So the thing to keep from this then is that in the analysis of imaging networks, of imaging data, we use convolution network, convolution layers, in addition to other layers that are used in regular, in general deep learning. So to remind you then, we have the radiomic, the feature analysis, and we have the deep learning analysis. And if you want to see how the another run at the feature analysis, we start out with the image. We identify regions of interest, and sometimes we need to also have the habitat of the tumor. We can render these as 2D, 3D. We extract features, and then we put them into machine learning algorithms and come up with analysis. And some of the key aspects of this work here is the segmentation, in other words, figuring out where the tumors are and so on, the definition of the features, and the classifier modeling at the end. So we have these two types of analysis. So now that you've become experts on how we do radiomics in imaging, I wanted to show you where all this is supposed to get to be useful, right? And that's in the analysis, in developing markers for radiomic, for imaging. So what do these markers do? They do the same thing that other imaging modalities are doing, and hence they have to be evaluated on the same metrics. For instance, question number one is the marker accurate? In other words, does it detect accurately? Does it predict accurately? These are two different kinds of tasks, which are sometimes conflated in the context of machine learning, but they're very distinct in the context of imaging, because diagnosis is the task of detecting a disease or an abnormality that is present, and prediction is the task of starting with a test result and predicting whether an abnormality will be there. So these are two different types of tasks in that go into the accuracy. Another one, once you have the accuracy question, is does the marker affect care? In other words, I and my graduate students can sit with a set of images and generate millions of radiomic features and markers and so on. Would they ever affect care, care if they were actually used? So this is the kind of question we ask about all diagnostic and predictive tests, and this is the kind of question that we ask about radiomic markers as well. Third is, do they affect outcome? So you can imagine that in order to find out if they affect outcome, you can put these in a clinical setting and do clinical trials with them. Or you can do simulations, like micro simulations and so on, and see how they might affect outcome. But the proof of the pudding is typically in the in being able to show that they affect outcome. So this complex machinery then that generate markers eventually needs to produce robust kinds of modalities, procedures, markers, if you want to call them, that will withstand this kind of an evaluation like any other diagnostic test. So the I will skip the slide. I'll just summarize it by saying most markers, which are radiomic markers today, are very much at the discovery stage. In other words, they're at the stage when we actually discover markers. Most of them are not ready for clinical evaluation and so on. Some of them are ready to be introduced, let's say, in a single institution study. And in fact, this is happening. Very few of them, if any, are at the mature stage, which are ready to be implemented in a multi-institutional setting. And even less are mature enough to go into the disseminated setting where we put them out on the internet, and I mean on the for very broad use. Although you'll see that although this kind of schematic has shaped the thinking in diagnostic imaging, that is not how it actually is perceived or in the world of machine learning. There's a lot of confusion about how do you go from here to here to here, and even more so, there's a lot of questioning as to why do you need to go from here to here to here. In other words, why don't you just go to the end of it and so on, have your app, and that's that. So I'll be making some of the argument that we need to think through this before we let some of these out there in the world, so to speak. And this is part of my a main concern of my presentation. So let's start from deep learning, and here is some recent examples. If you wanted to find the most recent stuff about deep learning for imaging, you can open the most recent issues of Radiology or Radiology-AI, Artificial Intelligence, which is the journal that is kind of focused on this. You could look at many other clinical journals that are starting to pick up these kinds of issues. But here's an example that came out in 2018. This was a paper that came out of a Japanese research team and was looking at staging of liver fibrosis using enhanced MRI images. This came out in April 18 in Radiology, and you could read the paper if you want. These are pictures of the liver, and this is an actual liver as it looks to the pathologist. This should be familiar to you from your kitchen, but these are human livers. The training set here was 530 patients. The test set was 100 patients, and the MR that was done was 1.5 to 3 Tesla. So these were the data. And this is a little more detail about the kind of pre-processing that you need to do when you have images like this. You cannot just take the whole image. You need to focus on specific areas and so on. You need to amplify them and crop them, and then you feed them into the neural network. And this is the picture about how the neural network analysis would work. You put in the input image that had 80 pixels. You do convolutions. Then you do pooling, and then you do some more convolutions. And then at the end of the day, the particular analysis was putting out, the last layer is called the fully connected layer for the image. The last analysis was putting out an index or a metric for fibrosis. So they took that index, and this is called the FDL score. DL, I assume, is for deep learning, okay? And they actually had a test set in which they tried to see how well you can detect with this score. And there was also a clinical classification of the specimens. And so they were specimens with cirrhosis, with advanced fibrosis, with substantial fibrosis, and so on. These would be the categories, and they dichotomized it and made ROC curves on the basis of the FDL score. And here is the area for the score. It was 84%, which is very respectable for any kind of diagnostic at this point, based on imaging. They also discussed sensitivity, specificity, and so on, and they looked at various other characteristics, various subsets, and various perturbations of the image. This is how that analysis went. So out of the main set of images, then there was a score was arrived at. It came out of the convolution network. And here's how this score, in a picture, here's how this score corresponds to the fibrosis stages. So it seems to track it fairly well. This is my quick summary then of what was there for the paper on fibrosis, and I use it as sort of one of the early examples of where somebody applied convolution networks to an image, and they came up at the end of the day with an index like this that seemed to track known measures of severity and so on. A couple of points here. When we get a score like this out of an image, the network doesn't tell you anything about the uncertainty of the score. I will come back to that, actually, because that's an important question. The other is, we don't know how reproducible this kind of analysis would be if we did it again, or if we did in a similar kind of images and so on. So reproducibility is another issue when it comes to measures and metrics that are based on radiomics, much in the same way as is an issue for any kind of imaging examination. So that's an application for a set of images, a finite, a moderate set of images, and a diagnostic or staging task. give you now another example that's more recent that was about lung cancer. This was lung cancer data from a screening trial. This is a screening trial that I was involved in. It was the National Lung Cancer Screening Trial, the NLST. And that study, that trial was comparing the impact of CT screening on mortality for people who were heavy smokers. So that study had about 50,000 patients, well, not patients, 50,000 participants. They were randomized to two groups. One group was screened with X-rays. The other one was screened with CT. This analysis is based on the CT data, and they published and they got the CT data on a subset on a subset of cases here, and they shown it to us, and they published it in Nature Medicine last year. They is a group from Google. They took all the CT images. They used both prior and current images. So they did one kind of analysis. Or they just used the current images because this was a screening study, which means there were serial images on an annual basis. They did the usual things about ROIs and so on. They also learned features and here is what they had as output. There was a probability of malignancy. There was a risk categorization. And then there was also another interest that they had was the localization of the cancer. So let's look at how successful that exercise was. And they compared their success to what human interpreters could achieve. So here is the prediction of malignancy, the ROCs for prediction of malignancy using the index that they had versus human interpreters. A bunch of human interpreters did the interpretation, and they gave them, they assessed sensitivity and specificity and so on. So the human interpreters were using a particular way of interpreting images called the lung rads and so on. And the circles here are the presentations of the human interpreters. This is just using the current. And then this one is using both the current and the prior CT. So part of the point was that that the Google people made is that this can do well against human interpretation. Obviously, that that's a strong statement, and it kind of, but the implication of it on the Google side is that this is ready now to use broadly on the, perhaps without human interpretation. So these things, in other words, the radiomic kind of modalities are real. They're here. They could be applied very broadly if they were evaluated appropriately and so on. And that's the bottom line. So I'm quoting here a piece of the discussion from that paper. It says this creates an opportunity to optimize the screening process via computer assistance and automation. Then that will show that the potential for deep learning models to increase the accuracy, consistency, and adoption of lung cancer screening program worldwide is substantial. So these are two examples of different kinds of studies in which deep learning was used to generate predictions and so on. And I will now go on to a third example in cancer. And after that third example, I will stop and take questions, because after that, we're going to go on into some feature space analysis. But the third example may be more interesting to biologists, but it is deep learning used to predict the time to event response. And what was and the images were not radiology images, but they were pathology images. So this was a study in brain cancer. They took their images from the TCGA, which is a database that has that are organized by NCI and has images, radiology images, pathology images, and clinical data for various kinds of cancers. So these investigators looked at, they were interested in how histology predicts, and they wanted to use deep learning for that. Now, the features for pathology images are a lot more complicated. The pictures are a lot more complicated versus what I was showing you before. Here are two regions of interest from histology slides. So they're fairly, the patterns are more complex, and obviously you need to work harder to have neural nets that will will work for you. But this work has also been done. I wanted to bring this example to you, one, because it links in with the analysis of radiology kinds of images, and also because in this particular study they looked at some correlation between radiomics and genomics, and this even has a name called radiogenomics. So I'll show you a couple of slides at the end of what they presented. It's also that this particular study is of interest for us at Brown, because we've taken these data and we're doing analysis about various ways of establishing methods for predicting time to event outcomes that take censoring into account. So what they did is the images they had 1024 by 1024 pixel images. They used a network that was already pre-trained. So if any of you wants to go very quickly and start doing analysis with imaging networks and so on, here's a pre-trained network called the VGG16, comes out of Oxford, and it wasn't trained on cancer data, on cancer images. It was trained, I think, generically, but it's somewhat trained already, so you don't have to spend a hell of a long time retraining it from scratch. You could start training it on your own networks, on your own data, and in fact, that's how this was done here. So they trained it. The output of each image of each analysis was a tensor of this dimension. And what we did at that point to see whether how the predictions would work is we took these and put them into a regularized Cox regression model, or we let the network go to the through its final layer, the fully connected neural net, and see what we got. So when we looked at the trained VGG16 and Cox regression, this is what we did. Okay, we took the final layer, all the features, we put it through Lasso. Lasso picked 28 features, and then we generated predictions. And the prediction we were trying to generate here was the prediction of survival past 914 days. And These here are box plots of how well we did. This is for the training set and for the test set. This is work that a student of ours, Samantha Morrison, is doing, and she has a paper on this. This is the same kind of analysis now, but instead of putting it at the end with the with Cox regression, so to speak, we used the fully connected network. Okay, and that one also did its combination and gave us predictions. I wanted to show you how accurate, how well the two methods did. And for that, we used the Breyer scores, and they were weighted to account for censoring. So it's kind of, performance is so-so based on these kinds of classifications. If you wanted to see a better, a more understandable, I guess, directly comparison, and so on, here are the predictions from the Cox regression here, the predictions from the fully connected network. Okay, they kind of track, but what we're doing with these data is trying to actually just use it as a test bed for various ways of estimating the predictive accuracy for this, because the data were censored. Now, I promised I will show you a little bit of radiogenomics analysis, and this is again in the same paper. This is sort of a recap of what they've done with the various, with the layers, with the slides. Okay, so and here are some pictures, some plots of what they came up with. They had genomic and histologic classifications of the gliomas. Okay, so they had astrocytoma, oligodendroglioma here, and here they had some mutations. Okay, and here they are showing how the prediction accuracy compares using various kinds of data and various types of features. So this is just based on histology. This is genomics. This is histology and genomics. And this is now with the neural network histology and another sort of variant of the neural network. Moral of this story is that you can combine neural net predictions and from images with genomic predictions, or you can see how the predictions from the neural network correlate with molecular subtype and histological subtype, which is kind of the direct correlation between what imaging says and what genomics says, and this is what's in this picture. So I'm going to stop here and ask for questions, and then we can proceed to the next step.
Thank you. So we have a few questions. So the first is, how does deep learning identify disease-relevant features in images for analysis?
That's a very good question, and the answer is very difficult. Deep learning doesn't tell you exactly what it does, and in that sense, it's a bit of a black box. And what people are doing is they go to the end, to the predictions, and then they try to work backwards to the image and see what kinds of features or what parts of the image would be driving the predictions you're making. this is an area of research in deep learning, and it gets to the question of exactly that you're raising. What's the face validity of what is being done? There's a lot of interest in this, but there are very few sort of usable generic approaches for this. In other words, there's no general algorithms for this. People are showing, okay, here's what I did. I took this image and I covered this piece, and then I went on and redid my predictions. And yes, lo and behold, covering that particular piece of the image affected the predictions and so on. But that's not a general theory. So you hit on a very good question here. And for us, for instance, when we did the regression with the final layer of all the features that came out of it, and these could be hundreds of thousands of features, and then we whittle it down, we don't know what these features are, because they came through this whole process of convolution and maximization, et cetera, throughout this. So that's the answer. I hope it's a real, it doesn't discourage you from going in and actually thinking about using deep learning. But this issue of face validity and what corresponds to what is a big issue and an item of research there.
So, and then somewhat along with that, they asked for censoring data. Did you simply, so this is with respect to the last example that you talked about, for censoring data, did you simply include the censoring status as a feature?
No, I don't know how the people did it at the end, but what we did is at the end of the day, when we had from the final layer, when we had the output of the final layer, we used we said that those were the predictors, so to speak, and the response was censored. And then we did more or less classical survival analysis kinds of approaches to do what we did.
Thank you.
Mm-hmm.
So, and then does deep learning require large sample sizes, and what sample size is large enough?
You know, that's another great question, right? People who train deep learning, they train them on these publicly available data sets, and many of them, they're not so many, but the ones that are out there, they're big, okay? In the clinical setting, if you're going to use deep learning, you cannot expect to have very large sample sizes. For instance, the Japanese study that I showed you as a first example had 800. And that's a lot. In the clinical setting, to come up with 800 livers, that's a very big deal. Okay. In the screening setting, where we had the images from the National Lung Cancer Screening Trial, these could be in the tens of thousands. So in that context, you may have large images, large data sets. But in the clinical setting, you have moderate numbers and both will do actually. You don't need, neither will you find sample sizes of 10 or 15,000 clinical images and corresponding truths, et cetera, to be able to train this. Okay, but it is fair to say that most of the uses of deep learning have been done using classifiers that were trained on large data sets, like what I showed you already, and I'll show you some more later on about how people trained data sets on apps, and so on.
Thank you. And then, let's see, are there concerns that the training data set will introduce bias in the outcomes when applied to test data sets?
Yes, and there is there's literature on this in the machine learning world. Some of that literature comes under the rubric domain adaptation. That's where you train a particular marker, a particular deep learning in one setting, and then you want to extend it to something else. And that's a very current area of research. That's a very difficult kind of question but it's absolutely relevant, because when the new machines, say even the new imaging machines that come into the hospitals, they may have a deep learning software at some point if the FDA approves it inside, which means it will have been trained in a particular kind of setting and a particular population. Then you bring it to the hospital in Indianapolis, that's a different population. And it's not just the prevalence, it could be that the types of, of abnormalities look slightly different. The other features in the image look slightly different, and so on. So there is a question as to whether a model that has been trained on a particular data set really applies to another data set. Some of that question is also the safety question. So the safety question is you can phrase it as follows. How do I know that if I take a deep learning modality and I bring it to a slightly new setting, how do I know that it's not going to go off the deep end in terms of its performance? And that is something that is not known. Theoretically, it's not known. It is not true that the performance, that what exists is bounds. Actually, I have a slide later on about it, but let's just talk about it right now. People have developed some mathematical bounds of how big the error will be and so on. These bounds are typically not very useful for practice. So it's an empirical question, actually, how you introduce a modality like this in any given setting and how do you monitor it. And in fact, the only kind of models, in a sense, or approaches that we have is how we monitor modalities that are used by humans. How do you evaluate? This means you need to keep evaluating it from time to time. You need to be monitoring how well it does and so on. And you may need to go back and retrain it, etc. That's the idea.
Thank you. We had actually gotten 4 of those questions kind of in the same category, asking about how generalizable the models are, all in a row. So expanding on that a little bit then, so how could we account for artifacts in the histology of the images? For example, folds in tissue, different H&E staining intensities, et cetera. And similarly then, if you do deep learning at one institution, would it work at another? So.
Well, I think I sort of addressed the latter, but the hope is that when you bring in your own images, you will train the network a little bit with part of these images, and then you'll start using it. In other words, you're not going to just take a machine, a network that was tested elsewhere and trained elsewhere, and you bring it back. But for artifacts, there's no way around it. In other words, what you see, I'm editorializing now, what you see in the literature are the success cases. In other words, you see the for instance, let's go back to these. Here are the images that they had, like the pathology images, right? Oh, these are nice and clean and so on, and etc. But if there was a little artifact here, it would throw off the thing. The hope was that the artifact doesn't represent something systemic. Otherwise, if the artifact is just some bad slide, so to speak, maybe that will wash out. In other words, not all slides will have it. So that's how you get through artifacts. But if all of the images from a particular hospital have the artifact, then there's nothing you can do. It becomes part of the analysis, part of the image, if they all have the artifact. So you have to be very careful here. And also remember that these, to reach images like this, as those of you who work in pathology, etc., you'll realize they've done a lot of pre-processing here to come to this state. It wasn't, it's not as simple, which means they cropped a lot, they left pieces out, they may have left cases out. So if you're going to take this and actually put it in a hospital for use, you have to have a very detailed understanding of how you will process the images, how you will develop, define the areas of interest, and so on and so forth. That's why when I started out with in one of the first few slides, I really tried to emphasize that the pre-processing of the image is very important for how these things would work. And what you see here are sort of the highest performance cases, because all of these things have been nicely done and they've been peer reviewed, blah, blah, blah.
Great. And yeah, and then similarly, how do you make sure, can you do batch correction or make sure that variation and staining from different batches won't affect the prediction?
Yeah. This is, that's difficult. It depends on how big the differences are between batches. Let's put it that way. If you went from one batch to another and you changed significantly the parameters of how you were processing the specimens, or how you were actually doing the imaging, then this can be thrown off because it doesn't know that it happened. So what you may observe is some kind of degradation in the performance. That's why monitoring of this in a real life setting is important. If you knew what the batches were, you could build them in as covariates. But then your model would learn these particular batches, which may not be useful when you take it out.
Absolutely, thank you. And then an attendee mentioned that it does, from what you had just said previously, sound as though it would also be important that the region of the image used is representative of the pathology.
Oh, absolutely.
Yeah.
Absolutely. For the that's definitely true for pathology, but it's also true about imaging. I mean, I'm talking about radiologic imaging. It depends how much of the tumor you actually bring into focus, and how good you are in segmenting. So the segmentation algorithms up front would be important for this, which is true for when humans interpret this. Depending on what they focus on, this could throw off their interpretation. It's just that here, this is a machine, so.
Yeah. Thank you so much. So you can, we made it through the questions so far, so we'll let you continue. Thank you so much for stopping and taking those.
Good. No, I'm very happy that these came up, because some of my later slides kind of get to some of these questions, but let's see if we can, we have another half hour, right?
Yes, over half an hour, yeah. Okay,
good. So let me see now how I'm going forward. No, I'm going backwards. Well, here is a slide I had about deep learning. I didn't get to it before the questions, but you can see that slide has some of the questions that you're raising, or at least raises issues very similar to what you're raising. For instance, the phase validity of the results. Okay. How do we know that these are real, so to speak, or what is driving them? What is driving what the deep learning is telling us? And so this is what I was saying there. There is a little area in machine learning called interpretable machine learning. In other words, how can we find features and characteristics of the source units that drive the outcomes of the. And as I was saying earlier, the methods that I've seen are ad hoc. There's no systematic way. Another question here, and that's very dear to statisticians, but not everybody else shares that concern, is the uncertainty of the results. Typically, people look at the outcome of machine learning, of deep learners, and they just treat the results as the outcome, no error bar around it. That's not true, okay? In the sense that there are various sources of variation that go into what deep learning tells you. On the one hand is epistemic variation, has to do with all the modeling and the assumptions that you have. On the other is there is variability and uncertainty due to the computation. At every stage for every layer for every one of the hidden layers, the model does an optimization and it calculates a set of parameters. These are very large numbers of parameters, hundreds of thousands. It does an optimization and then takes the output from the neural nets and goes from that layer and goes to the next layer. Well, there's uncertainty in that. So how do we account for this? Well, people have actually started to get to this, and it's a new area. I mean, you'll see papers in the last couple of years, and we actually have a couple of students who are working in that area from a statistical perspective. So some of the methods that are being proposed are what you might expect. There is people thought that they would go to say the final layer and put priors on the features and see if they can calculate posteriors. Now that's a lot of that's easy to say and very difficult to do because these are very large dimensional data and so on. So people are using very approximate methods, variational Bayes methods and so on to come up with calculations. That's what goes into Bayesian neural nets. Another one is dropout, where essentially you drop out subgroups of the cases and you try to use that approach to see how you can get estimates of the uncertainty. Another one is they call it deep ensemble, which I think it is just using several networks and doing model averaging that we call in statistics. But that's how they, that's another approach. So yes, there is work on how to assess the uncertainty of the results. And the first bullet here addresses the first question that was raised, namely the generalizability of the performance. So there are some sort of theoretical measures and theoretical constructs here. One is the so-called VC dimension of a model that has to do with the complexity of the model. And then there are bounds that take into account how complex is the model and how different, how divergent is the source domain from the target domain. Okay, this is this notion of domain adaptation. There's some elegant mathematical theory. Vapnik started it in the 90s and so on. In practice, at least all the bounds that I've seen are a little too big to be useful. And people, you'll see them being promoted in papers and so on, but I don't know of anybody who has a very, I'm not aware of any really useful versions of this that has been applied in practice. And definitely we need to know the answers of this before we start, at least that's my opinion, before we start taking some of these deep learning algorithms and putting them in actual use. You need to know how bad it can get, because there are real decisions that will be made of it. Pretty soon airplanes, if we ever get into airplanes again, airplanes are going to be flying using some of this. You don't want the bounds to be ridiculously large about that, right? So that's what I'm saying. We're scratching the surface in these questions, but that hasn't stopped people actually trying to use deep learning in real time. So with this, I will go on to the next. I had another example here from another deep learning thing. This was for cardiac disease, but I will not go through it. I will spend about 10 minutes showing you how we can do feature-based analysis with images. And for that one, I think many of you have seen high-dimensional data, or may have seen high-dimensional data being in biology and in areas like this. So that's why I only want to spend a short period of time on this. But here is an example. As I told you, in the feature analysis, you start out with a bunch of images. You use various kinds of principles and metrics and develop a whole bunch of features for this. And the whole bunch could be from very small to very large, depending on how much you want to do it. Many of these features are redundant. Many of these features are very correlated with each other. So then you have to whittle through them to come up with an answer. But the answers are more like anything you have seen in high-dimensional data. So here's an example. This was imaging of rectal cancer. And the question was to assess treatment response after neoadjuvant therapy. Notice here, we're not talking about very large numbers again. This is again a clinical setting. And now in the feature space, there were 141 patients. They were given neoadjuvant therapy. This was another paper from radiology. So many had PCR and so many had PR. And they did 2 kinds of MRI. But basically it was T2 weighted images that they were doing. They also did qualitative assessments. They computed, they computed 34 features. That's small. And they did a random forest classifier. So here are the features. I wanted to bring up the specific features just to give you a taste of what kinds of stuff people are looking at. They're looking at the signal, and they compute things like the energy, kurtosis, homogeneity of the signal, contrast, and so on. It's, entropy again, and there are other kinds of features that talk about how spread out the signal is, how concentrated it is, how intense it is, how it correlates with things around it. These are the kinds of features. So they used these features. They did as I was saying here, a random forest classification with these features. And at the end of the day, they came up with these. I mean, the goal was to predict PCR. They had here sensitivity, specificity. This was the sensitivity and the specificity of the MR features by themselves, and the qualitative method. And this is from the radiomics. Okay, and here's the area under the curve for radiomics. That's a very respectable ROC curve. So this was another paper that came out in radiology. There are lots of papers like this. Now, mind you, all of these features came out from a particular data set and so on. How well they will generalize is a question. And another question is, part of the generalization is, how reproducible are these features if you computed them again and so on? The answer is, it's not particular, they're not very reproducible. At least that's what I've seen in the literature. I'll give you an example here of another analysis that we actually worked with people from Colombia. We never published it, but I use it in my presentations. These were data from a study in which we were looking at renal cancer. So the question was, can we detect clear cell carcinoma? That's one of the bad kinds of renal cancer. And published a paper based on PET in JCO in 2013. And here are various kinds of images that go on. So some physicist friends at Columbia, okay, took the CT data for 190 cases, and they extracted 5,000 features. It's not 40 or 50, they extracted 5,000 features. And all of these features, you can group them into groups like size-related, first-order statistics, second-order statistics, shape, surface shape, wavelet features, fractal dimensions, Gabor energy, that's something physicists know, I don't, and so on and so forth. So they generated a lot of features. And as you can imagine, many of these features are very highly correlated. So they sent us these features, and we did some work with it. So we first did unsupervised clustering after features, ended up with 1,300 features out of the 5,000. Then we tried to get some more reduction and used lasso. Oops. For this. And then we did logistic regression with cross-validation and so on to be able to do this right. At the end of the day, we worked with 3 features. I mean, this seems a little anticlimactic, but 3 features worked reasonably well. In addition to that, we also did a random forest. And the random forest, as you can see here, the logistic regression from lasso was 0.77. The random forest was 0.8 in terms of the area under the curve. The logistic regression with lasso is a lot easier to understand. So the moral of this particular story is that, first of all, with these radiomics, we did reasonably well, actually comparable to what human observers did when they were interpreting these images. And then back to the point I was making about how features, how the world is in feature space. They need a lot of trimming. There's too many of them. So in particular, they may not be reproducible. And in fact, they may not be reproducible, and that has been shown in many cases. Others have shown that, well, you have to be careful that not all cases are like this. And this is now a paper here of, this is now a paper, the second paper is a paper actually written by our colleagues in Colombia that had generated the 5,000 features for us and showed that some features are reproducible essentially and others are not. But we should not be throwing out the baby with the bathwater here, that there is, there's a lot of information that can be gained. And I take their last sentence here as really take it to heart, saying you have to be careful about how you set the imaging acquisition parameters if you're going to compute features. A lot of the variability may come exactly from this difference in how you acquire the images and the lack of reproducibility. This gets back to the examples that people were saying about batches and so on. So that's as much as I wanted to say for this, and I will go on then to just revisit it and the question of how we evaluate markers. We're in the discovery phase. We're looking at reproducibility of the markers. We're seeking to optimize their performance. And all of the radiomic markers I know are pretty much in this space. But as I said, that's not an opinion that's shared by everybody. So let me show you some things that reflect opinion out there beyond, say, the very clinical realm. Machine learning in apps. Okay, you have all seen these apps now that you can get. You get the app that tells you whether you got melanoma or not. Melanoma. You put it you take a picture and so on. The app does some processing in the background and gives you some results. Alternatively, it may take your picture and beam it to a central place where it will give you the results. Now, mind you, I don't want to poo-poo this at all. This could be very much the way telemedicine is going to work. And in fact, don't be surprised that in the days that we live, that's a very important issue, even for the advanced industrial countries, like we used to have in the United States. We used to have industry. So, but if you are in a place where there's not a lot of medical infrastructure and it's concentrated in a few places and so on and so forth, this could be an important thing. So people are actually using these apps or are considering using these apps. So let me tell you what the experts in this area or are saying. Here's a paper from Nature that was looking at an app like this and dermatologist classifications of skin lesions. This came out in 2017 and essentially was saying this is a bright new world. We all should just get on it, on the bandwagon here. It says CNN achieves performance on par with all tested experts. And then it tells you all the things that are going to happen with 6 billion smartphones around the world and so on. We could really wipe out melanoma, so to speak, skin cancer. At the same time, they don't mention that with an app like this, and depending on where you set it and how many false positives it has and so on, you could inundate a healthcare system tomorrow. If we all take an app like this and start looking at our skin every day, there's no healthcare system that can really take care of this. So this is the downside of this kind of mass explosion of use of machine learning in imaging, and there are people who are fully behind it, like the folks in this article in Nature, and you can imagine there are people who are concerned. So here is some of the concerns, and they come from the professional societies. And some of the concerns say very few apps have ever been evaluated for clinical efficacy. And none has been sufficiently accurate and reliable using established research methodologies. This was something that was published in 2017 The situation may have changed a little bit. They were saying that there are no quality standards, there's no regulatory oversight, and then there are major ethical concerns regarding the confidentiality. When you use an app, the company that sold you the app has your data. And believe me, they can sell it anytime they want, and they are mining it as we speak. So when you use the app, you need to be aware of all of these kinds of considerations. And that's why it's sort of an area that we could move in gingerly. But all the issues that we discussed about machine learning apply here, and all the caveats and so on, but also all the promise. I mean, you have to balance the two. So we don't need to pay attention necessarily only to what Silicon Valley says about the promise, but there is promise out there. It's just that you have to walk a very careful pathway here. And in fact, the FDA has been trying to walk that pathway. So they have been regulating some of these devices. And for a long time, they wanted to regulate these devices as not standalone devices, but as devices that will be used by an expert. So, for instance, a couple of years ago, this was one of their first approvals in this space. There's a company called Arteris, and in addition to a software they had for detecting strokes and who should get an immediate, drugs for the stroke, etc. They had here an oncology suite. So they put out an oncology suite. The FDA approved it, but look at here what they say. The clinician retains the ultimate responsibility for making the pertinent diagnosis. This is the basis on which they approved it. So they say the oncology DL is a complement to standard procedures. This is where things were as of, say, a year ago. Things are moving, but I'd like you to pay attention to this statement by the FDA and related to some of the discussion we were talking about earlier about interpretable machine learning. Now, a clinician goes in, they have a certain, they take a look at things, they have some idea of what's going on. They use the machine learning software that gives them an answer. How can they check that the face validity of what they see? If this was a deep learning software, there are no checks today. So how could the physician responsibly make the decision by taking into account what the deep learner is saying, if the physician has no understanding of what's the face validity of what the deep learner is saying? That's a very important question. And I think a lot of people are obviously concerned about it. And the FDA in this one, in the sense that we're saying they were kind of, I don't know if you would say they were not taking a very strong stand. And this was realistic. They say, okay, you can look at it, but then you have to make your own judgment. Fine, if I have to make my own judgment, how would I go about making my own judgment in a principled way? If I don't know what the face validity, if I don't know what is driving the determination. So this is, there's not a lot of progress that has been made in that space. And this is where the interpretable machine learning considerations come in. So there are similar things about other applications of this. Here's one that is catching the interest of a lot of people. And actually, if any of you are into computing with found databases and participating in various kinds of contests about machine learning tools and so on, data that relate to diabetic retinopathy are out there. And they have been used in some of the standard competitions. So this is diabetic retinopathy data. And so, you take a picture of your retina. There's some quality assessment that is done someplace. The software can tell you its answer, or you beam it someplace, and then somebody can tell you their interpretation, which is, I'm sorry to say, which is how a lot of ophthalmology was done in the last 3 months. I mean, I know people who their physicians said, take a picture of your eye and send it to me. So this is not the future. We're there right now. So the FDA approved a device like this. And this device is actually, devices like this are starting to be used widely in places where there's not a lot of ophthalmology departments all over the place. For instance, I understand that in parts of India, devices like this are used, where you take images like this, you bring them, you beam them to a central place, and so on. With a device like this, obviously, if the device, if the message comes that you're okay, you're okay. If the message comes you're not okay, then that's sort of the limitation of telemedicine. Then you still got to do something about it, and that's not straightforward. Especially if this device has a lot of false positives. It will end up, for instance, inundating a system. It would inundate the local system in Boston. I mean, the Mass Eye and Ear. If we all had devices like this, we'll be banging down their door tomorrow. If these devices were said to be very sensitive. So that's part of what is involved here. But the FDA approved it as a screening device. So we're not in the future, we are there for some parts of it. The evaluation of these devices is kind of all over the place. In the clinical context, most of them have not been evaluated, but that's where we are. The FDA, as of recently, they were regulating these as computer-aided diagnostics, but that is not, they cannot hold on to this. Now they're starting to evaluate some of these as standalone things where they could make a recommendation. And then still clinical medicine, clinical healthcare and so on has to grapple with these recommendations. So we still have the same old questions: face validity of the results. What is the long-term property of the algorithms? Okay, one issue, and I'll finish with this. One issue that was an issue of contention was when the FDA approved some of these devices, they said, we approved the algorithm, the locked algorithm. In other words, the algorithm that is there today and is presented to us. If this algorithm continues learning, okay, this is now how, I'm speaking now, not the FDA. There's no theoretical guarantee that it's going to get better. It might go off the deep end. In other words, you might see a bunch of retinas that are very different and start interpreting everything the wrong way and so on. You don't know what the algorithm is going to do. So the long-term properties of the algorithm are not known. We don't have theory that says that these things grow, their performance is increasing monotonically or at least stay stable. It could go down depending on where you train them and what they see. Hence, somebody needs to know how we will monitor these devices in the future. And that is an issue, obviously, that radiologists and others have grappled with. How do you monitor how particular diagnostics and so on? Well, you periodically take a look at how well they're performing, et cetera, and do quality control and so on. All of these things have to be there. And I have another consideration here, which I think is pie in the sky. Under what conditions is the performance guaranteed to meet minimum standards? That's me speaking as a mathematician, if you wish, or as somebody who has been trained in, some kind of math. Can we specify some minimum theoretical conditions and so on? The answer is I haven't seen any. I think the answer is going to be very practical. That in the clinic and so on, we'll be monitoring these things much the way we monitor test interpreters and others. So I'll end then with this commentary. From the point of view of diagnosis and evaluating markers, we're faced with an avalanche of markers, which is an avalanche of riches, if you wish, but we still need to be able to sift through them. That's not a straightforward story. To put any of these markers to clinical evaluation, it takes a lot of energy, a lot of resources. And given that these markers are just based on various kinds of computer code, you could imagine that you could sit down with your graduate students and generate 20 tomorrow. And everybody can do that. So how do we sift through from the point of view of technology evaluation? How do we assess their variability when we start doing the clinical evaluation? How do we assess their reproducibility and so on and so forth? And finally, how do we check the face validity of this so that what the FDA says that the ultimate decision is in the hands of the radiologist, for instance, has actual, carry some water with it. Otherwise, it's just empty words because the physician doesn't know as much as anybody else. Why is the software telling them what to do if it's a deep learner? So that's all I had. And I had a list here of my collaborators who, Samantha Morrison has worked on these data on the REDX study, and she's working on the data with the pathology specimens that I showed you. John Steingerson is a colleague of mine who is an expert in diagnosis and prediction and survival analysis. And he is working with us. And Rotao Zhang is another one of our graduate students who is a brave soul. He's tackling a very difficult question. How do we assess uncertainty? And what practical use can we make of the uncertainty? We also have another student whose work I didn't feature here. That's why I didn't show. And she is looking into this problem of domain adaptation for markers that are based, that are developing ROC curves and so on. Her name is Bing Li. She's doing very mathematical work on that. So that's all I had. Thank you very much.
Thank you so much. So we have one more question so far. And that's, do you know how Google reproduces?
How Google reproduces?
Yeah.
Which Google? Which part of Google? Are you talking about?
I don't know.
Are we talking about the?
Word vec, or I don't know.
I have no idea about how Google reproduces, but I know. But in the paper I showed you, because this came out of Google for lung cancer, they didn't talk about reproducibility.
Mm-hmm.
This was a paper that came out of the Google empire, and so they didn't talk about reproducibility there. They were talking about putting machine learning to use on these models, on these data.
Okay. Let's see. Thank you. So are there any other questions? Here we go. So how sensitive would you expect feature extraction to site to noise variability? How sensitive?
That depends a lot on the feature, and it depends on the modality. It's a very good question. I think the physicists are trying to get a handle on it, but I can tell you that when it comes to looking at variations and calibrating, say, results from one scanner to another and so on. This is an ongoing kind of research. I think the radiomics is in a sense making all of this too precise, because it quantifies everything and generates very accurately computed, not measured, accurately computed features. But these features are really dependent on a lot of things. For instance, for years I was involved in a very simple kind of investigation. How does the standard uptake value of PET, how does it vary across scanners? And how can we calibrate the values of SUV from one scanner to another? That took a long amount of work for people to be able to sort of agree. And it's not like there is a simple formula that says if you took it from this scanner with this kind of tracer and so on, this is what you need to do. So you can imagine there, the SUVs are fairly robust. For those of you who have never seen an SUV, I'm sure any of your relatives who were in the hospital and they did a PET, they computed the SUV. It's the standard uptake value. It takes a region of the tumor and kind of averages the uptake of the isotope that you have put in, of the tracer. So you can imagine now, if it's an average over a particular part of the tumor, it depends on how you drew the contour in the tumor, how you did the averaging, and so on and so forth. And at what level of the data were you working? Because there is the level of the data that is at the picture that you see, and that depends on the reconstruction algorithm. But the real signal is different from the machine, and different machines, the signal is not the same, and so on. So if you're going to say anybody with an SUV change of less than X, 30%, means they haven't responded, well, you better be very sure. Well, we've been working for years now to be able to have a robust kind of answer to this. You can imagine now when it goes to the radiomic markers, which are a lot more finely computed. That's a very difficult thing. It will take a while.
Thank you so much.
Okay.
All right. Well, I think we're out of time. I'll give it back to Min. Thank you for an excellent talk and for answering so many questions.
And thank you for persevering.
Yeah, thank you, Dr. Gatsonis. Yeah, I would like to thank you all for joining us today. So our next talk will be this Thursday, and Dr. Mark Kelly from Indiana University will talk about translating the basic science discovery to new treatments for patients. So hope to see you on Thursday. Thank you.